Good morning and welcome to our webinar on overcoming crop stress and improving crop quality with Kelp Science Innovations. Some of you probably already familiar with me. I'm the presenter for this webinar. I'm Brian Cornelius, the Director of Applied Sciences here at Agrison. Not going to spend a lot of time on an introduction. Been in the agriculture industry for over 20 years, background in plant breeding. Now I'm working in these new biological or biocatalyst technologies, trying to figure out how to, one, design technologies and then how to show through data in the field the benefits of using these technologies. And again, today we're going to be focusing on technology in the kelp realm, some of the kelp-based technologies that are out there on the marketplace. This morning, we want to talk about, what is kelp and why is it important? I want to introduce the group to a new product called Maritime, offered through Loveland. Talk about why Maritime is different, look at some of the performance of Maritime, and then we're going to shift gears and get into another topic, talking about abiotic stress and how Maritime can actually be used to help mitigate some of those stressful conditions that we encounter, primarily the abiotic conditions. So, guys, first here, we're talking about why is kelp important for crop production? We've got, a lot of things out there. People call it seaweed or marine microalgae, but some of the important things are, they're rich in diverse compounds, lipids, proteins, carbohydrates, phytohormones, amino acids, osmoprotectants, antimicrobials, and other minerals. So these are the things that we're getting from, this kelp that we're running through some type of extraction method. And we'll talk a lot about the different extraction methods in this presentation. And then the benefits of those in agriculture. I mean, there's a lot of those. Stimulation of seed germination. We'll show you some information there. Enhancement of health and growth of plants, namely the shoot and root elongation, improved water and nutrient uptake, frost and saline resistance, biocontrol, resistance towards phytopathogenic organisms, remediation of pollutants, contaminated soil, and fertilization. We're going to talk about a lot of these things and show you information that proves that when you look at these technologies, it's probably worth your while to definitely give the Maritime technology a shot because it's doing a lot of these things that you see. Now, I just did a general Google search of the literature and found an article. You can see, in really small print down there from November 2016 from Journal of Environmental Science and Technology, just talking about what is kelp and what is it beneficial for. So we go from there to talk about this Maritime product. And if you're, familiar with naming conventions and all that, Maritime speaks directly to the ocean or water or something like that, because that's where we're getting this kelp source. Ascophyllum nodosum. It's just like the same source that most other products out there in the market are using. So when we get the question of how is it, different when you're using the same source, we'll get to that. It's very different because of the way we actually extract that material. But you can see those key features and benefits of the Maritime product. Improved soil microbial activity. We'll talk about that microbial community that's associated with it. Nutrient uptake, enhanced seed germination, early plant development, crop tolerance to abiotic stress. We're going to spend a fair amount of time in the second half of this webinar talking about abiotic stress. Highly soluble formulation that mixes very well with calcium-based or high-load fertilizer blends. Big difference from some of the technologies out there. We can get Maritime in any blend that you can put water in, which is probably 100% of those blends out there. We can put Maritime in those. We've got an OMRI listing and a WSDA organic certification for the product. So if you've got some organic production, Maritime is definitely a fit for that. So again, as we get into some of these innovative kelp sciences, when we talk about Maritime and the differences in Maritime and some other competitive products on the market, we've got a few boxes here that really speak to some of those differences. More active byproducts, we call them bioactives or biomolecules. namely primary and secondary metabolites for nutrient, increased nutrient and plant functioning, versus other kelp products that have a physical or chemical extraction. Chemical, primarily potassium hydroxide. Physical extraction, there's some kelp products out there that are a cold press or some other type of physical process. We've got a broad range of plant available phytohormone-like compounds. So when you look at the mode of action that we're going to talk a little bit about, we see phytohormone production or changes in the phytohormone production based on that the biological extraction versus a chemical extraction. Again, we talked about the ability to blend with fertilizers and soil amendments without sedimentation or precipitation or solid creating issues that can cause, strainers to be plugged or drip lines and things like that to have issues. And then you're going to see some results that show the positive results in ROI compared to some competitive situations out there. So when we look at that maritime production, you can see the picture. That's a picture, and that picture doesn't really do justice to the size of that system. We fit it into that picture, but that's a pretty large system there. But this is how we do our biological extraction. And again, we use the same Ascophyllum nodosum that most other seaweeds are using. So there's no question about the source. The source is the same, but the extraction method is very different. So we are taking a complete digestion of the kelp using a microbial or bioconversion of the kelp meal through a fermentation process. So again, very different. Now, what you get from that, you see this beaker, and you'll see another picture as we go through the presentation. This is what our finished product looks like. We get a very clean, amber colored material, no solids, no viscosity outside of, the viscosity of water. But that beaker is packed full of bioactive molecules extracted from the kelp, as well as a pretty robust microbial community that comes along with it. And we'll show you the benefit of that microbial community as well. So the first thing we did when we went down the road of developing a kelp-based product, when we were developing this maritime, we reached back and kind of looked at the history of our type of production cycles that we use here at Agrison, and we just took a different substrate. Now we're taking the kelp meal and we're creating this kelp-based product. And the first thing we wanted to do is do some type of test to tell us, is maritime like ordinary seaweeds or other competitive seaweeds on the market? So we did an FTIR, and FTIR basically gives you the functional groups associated with those products. And you look at these functional groups, maritime in green, competitive product A in red, competitive product B in blue, and look across that spectrograph. And you can see on the left side, things stack up pretty close. But as we get farther out towards the right, we see peaks that are higher, and we also see peaks that don't exist in the other products. So now this is telling us that, yes, the maritime product based on FTR looks like another seaweed, but our extraction method is getting things out of that kelp meal because it's a complete digestion that the chemical or the physical processes aren't doing. So this was our first indication that, hey, we've got a product that fits in this seaweed category, but we also have a product that has some differences, and some of these differences are pretty key when you look at it. So the next step, you can see those plus signs there, and you see those peaks that are either higher concentrations or peaks that don't exist in the other products. And then that takes us to the next slide that kind of shows you what some of those differences are. So we've got the bioactives based on the FTR. Again, the functional groups. You can see what the functionality of those bioactives is. But out to the right of that, you can see an X shows that those competitive products don't have that bioactive, and the check mark shows that Maritime does. So you can see we stack up pretty well against some of the competitive products from the standpoint of getting more out of that material than the chemical or physical extraction. So again, real key difference in Maritime is that it's a biological extraction that completely digests the kelp meal, and we get everything out of that kelp meal because there's nothing left. There's no solids left in the finished product. And then you can see what some of those functionalities are that we have because of that complete digestion. Now, I mentioned this earlier, and you'll see this coming up, is outside of having all of these bioactive molecules, we also have a microbial community associated with the product because of the biological extraction. You see 4 petri dishes here. You've got the total CFU counts, and then another key line on this table is the dilution rate. So you can see competitive product A basically has nothing. It's a 10 to the 1 dilution, which means it's only diluted one time. You go over to competitive product B, you can see the colony counts, but you look and it's all fungi. And we had to do a 2x dilution to get those colonies to separate. Competitive product C, you can see that it's a 3x dilution, and that product's mostly fungi. And then the Maritime, very diverse. We had to go out to a 4x dilution to get those colonies to separate. And again, very diverse. There's probably some fungi, bacteria, all of these other things in the Maritime product. So again, bioactive molecules, we find in more of those than we find in the chemical and physically extracted products. And then the diversity. We've got a lot of really nice, unique, diverse organisms associated with this. And the result of having a diverse microbial community is that you get a very diverse group of primary and secondary metabolites. So when we talk about that microbial diversity, this next slide is giving you a list of what some of those organisms are. And you can see there, I'm not going to try to read all of those because I can definitely tell you that I'll probably stumble over most of those. But what I will show you is that everywhere you see a check mark, you look at those functionalities or classifications of those organisms. We've got spore formers. We've got nitrogen fixers. We've got those that are associated with an ocean environment or a saline soil. We've got known plant-associated organisms. We've got known plant growth promotion traits associated with this product. So it's not just the bioactive molecules, it's also a very active microbial community that gives us that diverse group of metabolites. Now, why is that important? Why is it important to also have this diverse group of metabolites? Well, the next slide is going to give you the exact reason why. So these plates are designed, and you can see the Maritime on the left, competitive product A on the right. It has 31 carbon sources. It's a 96 well plate. It has 31 carbon sources replicated 3 times. And you can see that the Maritime, based on the information in this plate, is showing us that a metabolite or a byproduct produced as a result of that extraction allows 16 interactions, or there's something in that product that interacts with the sugar or carbon source on this plate. Competitive product A, if you look at it, just, initially it looks like, oh, wow, competitive product A has everything. That's just the color of that product that stains the plate. There's no interactions with that product A. Now let's look at competitive product B. Different story. Now this one really shows you that there's nothing happening with competitive product B. And then let's go to competitive product C. Same deal. There's no interactions, because if you remember the table that showed the microbial population, one of those products had nothing, one of them had all fungi, and the next one just had mostly fungi, where Maritime had a very diverse community of bacteria and other types of organisms. So guys, we're talking about the differences and why that's important. The next thing we want to look at is, after we see the differences in Maritime and how we extract it and what we get out of our process, we want to talk about what's actually happening with the plant. So now we're getting at, okay, let's figure out what the mode of action is. So we see that initially, some of the work that we're doing, that there's some phytohormone production changes in the plant. Uh, and you can see the way we do that is we've got a treated plant versus an untreated plant. We wait about 5 days, we harvest those plants, we extract the RNA or DNA from those, look at the sequences, and then do some genome analysis and see what happens within the plant. What genes are upregulated, downregulated, what changes are happening in that system. And then we'll show you a graphic of that later on as it relates to some abiotic stress conditions in the field. But you can see here that we're seeing, some downregulation of auxins and IAA or indoleacetic acid. And then we're seeing some upregulation of cytokinins and things like that. So now we're trying to dig deeper into what's the mode of action? What's the active site that these bioactive molecules are, really doing something in the plant? So this is some of the future work. Now this is some work it takes a while and it's pretty expensive to do, but here we're committed to fully understanding what's happening with these technologies and getting that information out to you guys so you have a better sense of, if you're going to use this technology, how is it going to affect your production? So those are some of the things that we're dedicated to figuring out, and we're just scratching the surface on some of that and some of the work that we're doing at our science company and some of our partners out there in the marketplace. Now, let's kind of get back to it. Again, we want to talk about the differences. So here we see what happens, or what we feel like is happening with some of the biologically extracted materials versus the chemically extracted materials. So you can see that table on the left side is chemically extracted versus biologically extracted on the right. We talked about that microbial community. It either has it or not. And again, on the maritime side, very diverse community. If you don't have a microbial community, you're very limited to the amount of secondary metabolites that you can have. And with the chemically extracted products you saw, they were either all fungi or mostly fungi. So the only metabolites you could get are those produced by those few fungal organisms. Salt or salinity stress relief, where you may get some with the chemically extracted ones, we see some very superior results with the biological extraction. The chemical extraction, from our experience, gives you a lot of undigested or unprocessed material at the back end of those chemically extracted processes versus the complete digestion with the biological process. We feel like that chemical extraction may also damage some of the phytohormones that are in that material versus a very, very clean process with the biological extraction to get more of those out. So again, more about the differences. So now we talked about the differences. What does that give us? It gives us a lot of versatility. So when you look at Maritime and you look at how we can use that product in our nutritional programs, it gives us a lot of flexibility where we can go into specialties: grapes, citrus, strawberries, cereal crops, legume crops, turf and ornamentals, tree crops. We're not limited to where we can put this product. We're not limited to just a foliar application versus a soil application. We can go in almost any situation where you're putting something out in the field. And here's a big key. One of the things that we've learned is that, especially out west, calcium-based fertilizers are huge. And one of the limitations with some of these other products, where the pH may be high or they may be extracted with potassium hydroxide, we're very limited in how much of that material can go into those blends, especially if those materials are being applied as a foliar. So again, we wanted to make sure that this technology checked all the boxes for versatility and the ability to go in applications where a lot of these other products are very limited. So here's one of the big keys that we learned, or that we were told when we were developing this technology. We need something that we can actually put with CAN 17. So you can see this table in front of you, going left to right, is increasing percentages of CAN 17 from 10% up to 100% CAN 17. On the other side, going from right to left, you can see increasing percentages of Maritime. So from 0 to 90%. And you can see that in all of those, they hold up very well. There's no issue getting Maritime into these CAN 17 or calcium-based fertilizer blends. Like we said earlier, if you can get water into a fertilizer blend, you can get Maritime into that blend. So no issues there. All right. Now, I talked about this a little bit earlier, but I want to show you just a little graphic of how this works when we start talking about stressful conditions. We were working on, some of the transcriptional information of, what's actually happening in the plant in a general sense. We spray with the product. We let that sit for about 5 days. We extract the RNA and DNA and we sequence that and we look at what's happening inside of the plant, generally under ideal conditions. But now we want to go to a more stressful condition. So what happens under abiotic stress conditions, where there's heat, drought, salinity, other environmental conditions, cold, flooding conditions? How do we see what's going on with the plant when we have the product associated with that? On the left side, you see in an untreated condition, you look at that little circle there and you see what's happening with the plant from a genetic standpoint. These are the genes that are actually functioning. You can see different colors there. On the right side, you can see if you split that plant in half, we've got a treated plant with Maritime. You see better growth and development, but why is that happening? You can see that there's different sets of genes that are functioning or different genes that are turned on, upregulated, downregulated, even down into the root system. You can see where there may be different root exudates that are being produced. This is what we're looking at when we talk about stressful conditions and applying the Maritime product to these stressful conditions. We're trying to figure out so you guys are better educated on how this product works and what's the specific mode of action is under these conditions. Now we're going to jump into a few data slides and we're going to talk about, some of the things that are happening under ideal conditions, and then we'll again get into some more stressful conditions. So we started developing the Maritime product in the summer of 2016. Pretty quick, we were able to get the system to a maturity where we could actually put some of that product in the field. So I took the product to Yuma, Arizona in a replicated trial that we harvested in early 2017. And the first trial in the field was head lettuce. And you can see some of the results from that trial where we used a competitive product in the marketplace at 2 quarts versus the Maritime product at 1 and 2 quarts. So here's the data from the marketable percent of lettuce heads that came out of the field and the actual yield. So you can see 71.1 with the competitive product, 75 with the 1 quart, 76 with the 2 quarts of Maritime. And then we go over to look at the yield, 20 tons with the competitor, 20.9 with the 1 quart, 23.3 tons with the 2 quarts. And at that time, at those market prices, that 23.3 tons was about $900 more per acre to that grower. So again, significantly increased the marketable percent as well as the total yield compared to some other products. Now again, this is a commercial situation, very little stress, pretty ideal conditions. We go to 2018, we're repeating some of this work, and now we're doing some work at the University of Arizona at their research farm. Again, everything is commercial production. The only part of that crop that's harvested is crop that meets marketable quality. So again, grower standard practice, 21.3 tons and another 5 tons with the Maritime at 2 quarts per acre. You can see over there, the picture on the right, the grower standard or no Maritime versus treated with Maritime. So there's some other differences there. We had some differences in chlorophyll readings and things like that. So lots of information coming from the field. So look here, we're just looking at Maritime versus the grower standard. This next slide that you're going to see, we've got 2 competitive products in that same trial. So now you can see how those stack up. Now, The competitive products increase yield, absolutely, but not to the level of the Maritime. So very, very good results in Yuma in 2018. Looking at boxes per acre, if you're looking at that, which is what a lot of you guys, if you're producing lettuce, you get paid on number of boxes. Again, pretty good results with the Maritime. And like I said earlier, not knocking the other competitive products, but we're just trying to show you the differences when we get those products in the field compared to the Maritime. And lastly, let's look at the dollars. That's a pretty big difference there. More than double the competitive product A, almost double competitive product B. That's a pretty significant increase with 2 quarts of the acre run through the drip on some head lettuce. Let's go to 2019. This is data that we just got in here recently. Now we go to the coastal part of California looking at celery. And if you look at this, you can see there the grower standard versus Maritime. There's some pretty good differences there. Look at just the sheer above ground biomass that's being harvested on the celery, the number of shoots and the thickness of those. That's a pretty good difference. So we look at the root biomass, you can see again, Maritime's got the advantage. And you saw the picture showing that increased above ground biomass that's actually being harvested. So again, guys, we're just trying to show you information from the field, looking at it in commercial situations. Maritime definitely has a fit. Now, as we shift gears, let's start talking about abiotic stress. Now we get into some of these fields where we might have some challenging conditions, whether it's drought, salinity, heat, cold, flooding, nutrient deficiencies, all of these things that are non-living or environmental factors. How do we try to help mitigate some of those things that we really don't have much control over? Even when we can, control irrigation, heat can still be a problem, salt and things like that. So we're trying to use this as a mitigation for some of those problems. And these problems, if you look at it worldwide, are pretty costly. There's a lot of money lost every year to abiotic stress conditions, and we'll see some of that information as we go along. Let's take a quick look back. Let's look back over the last 50 years. And if you look at some of the conditions over the last 50 years that have caused yield losses to our growers in the US, we've got, blight way back around 1970, but almost every major event since 1970 over the last 40 years has been an abiotic stress condition. Wet, early frost, drought, flooding conditions, and then some other unusual climatic events. So guys, this is what we're talking about. Most of our major yield losses are due to abiotic conditions. You see a couple of pictures there. That's corn under drought, and you can see some wheat under drought conditions. Guys, salinity, this is a major problem around the US as well as around the world. So let's look at some of those conditions and how that affects us. 75 countries are affected by salt degradation around the world. 20% of irrigated land is also affected by this, and that leads to over $27 billion just to that one condition, salinity. So now let's add drought, cold, flood, heat, all of these other conditions combined. That's a pretty big number if we've got just 27 billion from one of those conditions. Look at some more numbers. Again, 26% of the US crop land is affected in any given year. If you remember back just months ago, how many of you guys were affected by the excessive wet conditions in 2019? We've got a picture in our office showing a tractor pulling a planter, but the tractor is like a boat and the planter is going across the water. That's what a lot of 2019 looked like. If you were in the Midwest in 2012, it wasn't flooding, it was drought. So 26% of the U.S. cropland in any given year is affected by drought. 38% globally affected by drought. And that's 70% of global production uh, is fed from these acres. That's a lot of our production. So 67% of total crop losses in the US in the past 50 years by drought. So you add that to 27 billion from salinity. Guys, we've got a big issue with abiotic stress, and we're going to show you some ways that we can try to mitigate that with some of these new technologies. So, abiotic stress and yield. How are we affecting the plant when we take some of these key kelp-based technologies out there? Looking at the whole plant on the left side of that, the reactive oxygen species scavenging, that's going to be a way we can help mitigate some of that, helping improve our membrane stability, helping improve our osmoprotection. Guys, from a whole plant standpoint, we can do these things. Now, if we break that into root and shoots, stomatal regulation, xylem, hydraulic conductance, thicker stems in the shoots. In the roots, if we can improve water availability, ethylene and oxygen levels, and actually give us deeper, more vigorous roots, these are some of the things or some of the ways that we can actually help mitigate some of these abiotic stresses. Let's show you an example of the differences when we actually artificially introduce drought stress in some of the plants. So you can see here, this is some work that was done by our science company, Acre Sciences. They do a lot of really fantastic work for us. We've got some radish here, and you can see on the top, that's an untreated, and on the bottom, those plants are treated with Maritime. Look at that arrow there that shows the last day of water. And as we go out from that, the untreated control crashes pretty hard. I don't see any differences in the Maritime treatment in that time period. Not exactly sure what that time period is, but you can see what we're talking about. Drought stress, Maritime really affects those plants under drought conditions. So let's summarize everything we've talked about over the last 40 minutes or so. Again, it's that extraction, biological versus chemical and physical. We see some differences there. Very clean product, complete digestion. Broader range of benefits versus other kelp-based technologies. We're seeing that in furrow, drip irrigation, foliar applications. We're seeing that we can put this in a lot of different places. Stress relief, salt, abiotic. We just talked about that in this last section. Plant growth promoting microorganisms. We've got those in there. Those guys are giving us some benefits. We have the ability to get this into organic production. We've got an OMRI listing. We've got a Washington State Department of Ag organic certification, and we've got that superior handling and compatibility to go along with that. We really appreciate you guys joining us this morning. Have a good day.